Assalamu alaikum. My name is Hajar Yunus, and in the chapter of cell, the unit of life, we will discuss about endoplasmic reticulum, another organelle of cell. So, endoplasmic reticulum is also abbreviated as ER. E represents endoplasmic and R represents reticulum. So endoplasmic reticulum was observed by Garnier. Means Garnier was the scientist who told that there was an organelle which was called endoplasmic reticulum. And ultrastructure was observed by three scientists, and these three scientists were Fulens, Claude, and Porter. And what is ultrastructure? Ultrastructure means what is present inside the endoplasmic reticulum when the endoplasmic reticulum is observed under the electron microscope. So three scientists observed the ultrastructure, and the name was given by Porter. So endoplasmic reticulum, in which cell endoplasmic reticulum is present or not. Firstly, endoplasmic reticulum is the membrane bounded organelles, and as you know, membrane bounded organelle is present in eukaryotic cells. So endoplasmic reticulum is present in eukaryotic cell because eukaryotic cell is the contains the membrane bounded organelle, whereas endoplasmic reticulum is absent in prokaryotic cell, because in prokaryotic cell, as you know, that prokaryotic cell does not contain the proper nucleus, and the proper prokaryotic cell does not contain the or does not have the membrane bounded organelle, and endoplasmic reticulum is the membrane bounded organelle, and it is absent in prokaryotic cell. Next is endoplasmic reticulum is present in eukaryotic cell, but endoplasmic reticulum is not present in mature mammalian RBC and germinal cell. So next is the element of ER, means which elements are present in endoplasmic reticulum. So in endoplasmic reticulum, firstly is the cisternae or cisterna is present. Second, vesicles are present, and third, tubules. Means endoplasmic reticulum contains three elements which are cisterna, vesicles, and tubules. Firstly is the cisterna. Cisterna is formed by protruding out from the nuclear membrane. So this is the nucleus, and here the nuclear membrane is present. So this nuclear membrane protrudes out and form this flattened structure, and this flattened structure is stacked on each other. In this diagram, you can also see here the nucleus is present and outside membrane is present, and the nucleus double membrane is present, and here the nuclear membrane protrudes out and form this flattened structure, and this flattened structure is called the cisterna. This cisterna is studied with ribosomes. As you can see here, this red colored structure, these are the ribosomes on the cisterna. Ribosomes are present, and these ribosomes are attached with the help of riboforeign protein. Means these ribosomes are present, and these ribosomes are attached with the help of something or some protein, and in that case the protein is riboforeign protein. Next is closer to the nucleus, and means this cisterna is closer to the nucleus because this structure, this cisterna protrudes out from the nuclear membrane, and this clearly shows that cisterna is closer to the or nearer to the nucleus. And next is unbranched structure with the 40 to 50 micrometer lumen, and this is not a branch structure, this is the unbranched structure. And here the diameter of lumen, means what is lumen? Lumen is inside inner structure, or of the or inner uh, inside of the cisterna. So the diameter of the lumen of cisterna is 40 to 50 micron meter. So the first element was cisterna, and let me explain again that cisterna, firstly cisterna protrudes out from the nuclear membrane, and it is the flattened structure, and they may or may not be connected to one another, and on that ribosomes are present, and ribosomes are studied with the help of protein, and the protein is called riboforeign protein because this structure is or this structure protrudes out from the nuclear membrane. That's why it is closer to the nucleus and it is unbranched structure. Next is vesicles. Vesicles are closer to cisterna. As you can see in this diagram, in here, these are the vesicles, and these vesicles are closer to cisterna. Round in shape, these vesicles, as you can see, this vesicle is round in shape, and the lumen is 50 to 500 micrometer. So the diameter of the lumen of vesicle is 50 to 500 micrometer. And the last element of endoplasmic reticulum is tubules, and this is the this structure is the tubule. This is branched. As you can see, this is the branch structure. There is a difference between the cisterna and the tubules. Cisterna is the unbranched structure, whereas tubules are the branch structure with a diameter of 50 to 100 micrometer lumen. And next is this tubules are closer to the plasma membrane. As you can see, if this is the cell, and here the nucleus is present, and here the so here the nuclear membrane protrudes out and from the structure, and this flattened structure structure is connected to one another, and here the branch structure is present, and this branch structure is actually the tubules. And you can say this is the plasma membrane, and here the tubules are closer to plasma membrane. That's why I have written that plasma tubules are closer to plasma membrane. So let me summarize this slide. Elements of endoplasmic reticulum are cisterna, vesicles, and the tubules. And the cisterna is the structure which protrudes out, and the nuclear membrane protrudes out from the cisterna, protrudes out from the nuclear membrane, and the vesicles are closer to cisterna, and the tubules are closer to plasma membrane. Next is the types of endoplasmic reticulum. There are two types of endoplasmic reticulum. One is smooth endoplasmic reticulum and other is the rough endoplasmic reticulum. So smooth endoplasmic reticulum is near the plasma membrane, and it is called smooth endoplasmic reticulum because this structure is not studded with ribosome, and you can say nothing is present on the smooth endoplasmic reticulum, and that's why it is called the smooth endoplasmic reticulum. Whereas in case of the rough endoplasmic reticulum, in the last slide we studied that, or we learned that near the nucleus, the structure is present which is studded with the ribosome, and this structure is called the uh, cisterna. And actually this cisterna is the part of the, or this cisterna is the rough endoplasmic reticulum, because in case of rough endoplasmic reticulum, ribosomes are present. So first point, smooth endoplasmic reticulum are mainly present near the cell membrane, and rough endoplasmic reticulum are present near the nucleus. Second is no ribosomes are present. That's why it is called smooth endoplasmic reticulum, whereas in case of rough endoplasmic reticulum, ribosomes are present. That's why it is called the rough, because something is present because of which surface is rough. That's why it is called rough endoplasmic reticulum. Smooth endoplasmic reticulum are mainly present in lipid forming cell, or you can say smooth endoplasmic reticulum plays a role in the lipid formation. That's why these are present in large number in lipid forming cell, for example adipose cells, leukocytes, etc. Whereas rough endoplasmic reticulum has the ribosomes. That's why they are mainly present in the protein forming cells, for example Nissel's granules and echinal cells. uh, Fourth point is smooth endoplasmic reticulum are originated from the rough endoplasmic reticulum. If we talk about origination, rough endoplasmic reticulum are originated from nuclear membrane, means in case of the rough endoplasmic reticulum, nuclear membrane protrudes out and form the rough endoplasmic reticulum, and on that ribosomes are present. Whereas in case of smooth endoplasmic reticulum, after some time rough endoplasmic reticulum loses the ribosomes, and now ribosomes are not present. Now this structure is called the smooth endoplasmic reticulum. That's why smooth endoplasmic reticulum are originated from the rough endoplasmic reticulum. Smooth endoplasmic reticulum are mainly formed by the tubules, In the last slide, we saw that near the plasma membrane, tubules are present, and tubules are branch structure, and that's why smooth endoplasmic reticulum are mainly formed by the tubules, whereas rough endoplasmic reticulum are mainly formed by the cisterna. So these are the differences between smooth endoplasmic reticulum and rough endoplasmic reticulum. In this slide, we will discuss about functions of endoplasmic reticulum. So first function of endoplasmic reticulum is endoplasmic reticulum gives framework to cell or acts as cytoskeleton. And how endoplasmic reticulum acts as cytoskeleton? As you can see in this diagram, here in the center nucleus is present, and here the rough endoplasmic reticulum is present, and here the smooth endoplasmic reticulum is present. So it uh, acts as a cytoskeleton, because of which or because of this endoplasmic reticulum the shape is given to the cell. So that's why it is called endoplasmic reticulum gives framework to cell or also acts as a cytoskeleton. Next is acts as connection between nuclear membrane and plasma membrane, and it's true because here the near the nucleus cisternae are present, and near the plasma membrane tubules are present, and these are connected because the origin of rough endoplasmic reticulum is nuclear membrane, and origin of smooth endoplasmic reticulum is the rough endoplasmic reticulum, and they are connected. That's why this endoplasmic reticulum acts as a connection between nuclear membrane and plasma membrane. Third point is endoplasmic reticulum has about 30 enzymes which take part in biosynthesis. In the membrane of endoplasmic reticulum, 30 enzymes are present, and these enzymes take part in the biosynthesis of the cell. Next is produce and transport lipids and proteins to Golgi body. So here the function of endoplasmic reticulum, and here the rough endoplasmic reticulum is present. So the function of rough endoplasmic reticulum is protein synthesis, and here smooth endoplasmic reticulum is present, and smooth endoplasmic reticulum does not contain ribosomes, so they take part in the uh, lipid synthesis. So here in the endoplasmic reticulum, protein and lipids are synthesized and are transported to the Golgi bodies for modification. And for the Golgi bodies, we will discuss about Golgi bodies in other slide. Next is endoplasmic reticulum helps in the compartmentation, and how endoplasmic reticulum helps in the compartmentation, because here this structure is present, and because of that there are the compartments are present. That's why endoplasmic reticulum helps in compartmentation. Next is cell plate formation, which is also called the phragmoplast. What is phragmoplast? Phragmoplast is the pieces of Golgi body and ER, and these phragmoplast helps in the cell plate formation in case of plant cell. Plant cell, like in case of the cell cycle in the plant cell, after the telophase there is a process of the cytokinesis, and in case of cytokinesis cell division takes place. In case of animal cell there is no problem, because in case of animal cell wall is not present. That's why the structure is not very rigid or not rigid, whereas in case of plant cell wall is present. Because of cell wall the structure is rigid, and that's why here the cell wall does not let the plasma membrane or even the cell wall does not move or does not invaginate inside, and two cells are not formed. So in case of the plant cell there is a phragmoplast, means the pieces of endoplasmic reticulum and the Golgi bodies arranged in the center, and in that manner two cells are formed. So here the endoplasmic reticulum is, or endoplasmic reticulum takes part in the cell plate formation or phragmoplast. Next is the GERL system. Endoplasmic reticulum is the part of GERL system. G stands for Golgi body, R stand for endoplasmic reticulum, and L stands for lysosome. So GERL system, endoplasmic reticulum is the part of GERL system, and this GERL system is very important. And how GERL system is very important? Because this is the cell, and here this is the Golgi body. Here we will talk about GERL L system. So here the G means Golgi body. So here the Golgi body is present, here endoplasmic reticulum is present, and here the lysosomes are present. So how, if outside something is present, like here the food particle is present and something is present, so firstly here the vesicle is formed, and vesicle is the cup shaped structure. So here the plasma membrane forms the cup shaped structure, and here the food material is taken in, and after that in the here this is the vesicle, and now here this is the lysosome, and in the cell lysosomes are present, and in the cell if the lysosomes are not working, then they are called the primary lysosomes, because they are inactive at that time. When their vesicle is present, then this primary lysosome attaches to the vesicle, and now we start calling it secondary lysosome, and this secondary lysosome is active. So if there is a question, which one is the active lysosome and which one is the inactive lysosome, so primary lysosome is the inactive lysosome and the secondary lysosome is the active lysosome. So here after that this lysosome contains the hydrolytic enzymes. Many hydrolytic enzymes are present. So firstly here the wall is dissolved, and after that inside, with the help of the hydrolytic enzymes inside, the food materials or whatever is present is digested, and this material is absorbed by the cell. So firstly there is a process is formed by the cell. So there is a process how lysosomes are formed. So here the endoplasmic reticulum is present, and this lysosome is the, or lysosome contains the hydrolytic enzymes, means the protein is present. So here the endoplasmic reticulum, or mostly the smooth rough endoplasmic reticulum, takes part in the protein synthesis, and after the formation of the protein synthesis, here the vesicles are present, and this protein is packed inside the vesicle and is transported to the Golgi bodies. And here some modifications are taken place or are carried out, and after the modification, the product which is dumped outside the Golgi body is the lysosome, and how the lysosome is formed. And after the formation of lysosome, the food material or whatever is outside is taken in. This lysosome attaches to the vesicle, and after the wall is dissolved and the material is digested and material is absorbed, and after that the undigested food material, and here you can say this is the undigested food material, this is the undigested food material, this undigested food material is dumped outside the cell. So how the GERL system, and in case of the GERL system, the vesicle is formed, and how the vesicle is formed, vesicle is formed by the invagination of the plasma membrane. So firstly vesicle is formed means small part of cytoplasm is, or small part of plasma membrane is not present. So if and this part of the plasma membrane is given back when the undigested food material is dumped outside the plasma membrane. In that case this vesicle is attaches to the, or this vesicle attaches to the plasma membrane, and the undigested food material is dumped outside the cell, and again the again plasma membrane becomes the part of or again small part of plasma membrane becomes part of its um, is become part of its become its part. So every time small piece of plasma membrane is not available, so after some time if this piece of plasma membrane is not given back, then after some time the surface area of plasma membrane will be less. So GERL system is very important. If I summarize this slide, in case of the GERL system, firstly the G means endoplasmic reticulum forms the protein is packed in the vesicles are transported to the Golgi bodies modified the protein, and after the protein modification the material or the material is packed and is packed in the Golgian apparatus or Golgian bodies, and is uh, transported in the cell, and now this structure is called the lysosome. Now the lysosomes are present in the cell, and if the cell wants to take in, or something wants to enter the cell. Now here the vesicle is formed, and this vesicle is formed by the invagination of plasma membrane. Now the vesicle is formed, and after going through all the step, now the undigested food material is present on this undigested food material needs to be transported or needs to be dumped outside the plasma membrane. In that case, the vesicle attaches to the plasma membrane, and now what is or what is on is given back to the plasma membrane. And now in that case, this is called the GERL system, and this GERL system takes a very important part in the uh, uh, in, takes a very important part. If the small amount piece of the plasma membrane is not given to is not given back, after some time the plasma membrane will be less. In that case, surface areas will be less, and at the result, and it results in the or it results in the disturbing the functioning of plasma membrane. Thank you so much.